Hi, good morning again, and thank you for joining us for this second session of our webinar today. And the first speaker of this session will be Shilpa from India. Please.
First of all, good morning everyone, and myself Shilpa Prabhakara. I'm from India, and my research exercise topic is identifying viruses causing yellow leaf curl disease of tomato in Israel. I did my research exercise under the supervision of Professor Hanu Kaznuk. Background information about TYLCV is tomato yellow leaf curl virus is one of the most destructive viral disease almost the worldwide, and it causes 100% yield loss in many countries. The yield loss will be depend on the stages which the plant is going to affect. If it is affected in early stages of the growth, it will cause 100% yield loss. If it is later stages or like vegetative growth stages, it will cause 30 to 40% of yield loss. The TYLCV from Israel was the first virus characterized, and diversity of TYLCV viruses we can see in Mediterranean basin and Middle East centers. The TYLCV virus belongs to the genus Bigomoviruses and family Geminiviridae. The source of infection is whiteflies. It's called Bemisia tobacci. It will affect all stages of plant growth like seedling stage, generative stage and vegetative growing stages. It will affect all parts of the plant like leaves, stems and whole plants. And symptoms of affected plants like leaves will stunt and bushy growth, reduced size and abnormal forms. Flowers will drop after the infection, and stems will grow abnormally and whole plant will drop. This is a These are the images showing how the infected plant will look like stunty, dwarf and abnormal conditions of the plant. And the adult stage of the whitefly will transmit the viruses from plants to plants by sucking the sap from the plants. These are the egg stages and larval stages. These will not affect the plant, but adult stage will transfer the viruses from plants to the another plant. The sequencing as indicated that TYLCV includes a large number of viruses and strains. The sum of the viruses here I mentioned are named based on the region where they find, like Israel, mild from Jordan areas, and Sudan, Malaga, Sardinia virus and Mali viruses. My study objectives are finding the different TYLCV associated viruses belong to the same genus which causing tomato yellow leaf curl virus disease in tomato in Israel, and worldwide geographical survey of TYLCV viruses by comparison of sequences of TYLCV nucleotides and proteins, code proteins. According to my first objective, the materials and methods are the first step is sample collection. The symptomatic leaf samples were collected from Rehovot and Betagon. Those will going to keep at -80°C until we use. A nucleic acid extraction, we are going to extract a DNA from the infected plants and normal plants by using dactylifera extraction method described by Potter. Second step is primer designing and PCR reaction. The virus-specific oligonucleotide primers were used to detect TYLCV in symptomatic samples collected in Israel. We are here interesting three viruses that are TYLCV-ES, TYLCV-IL and TYLCV-mild. The set of primers I am going to show in next slide, and parameters for the PCR reaction we are going to set. This is the figure is showing that where the locations of the primers and the viruses and virus genome and expected lengths of the PCR products. The primer location is commonly in introgenic region is called IR, and the expected lengths of base pairs is depend on primer sets we are going to use for each virus. This the table is showing that which are the primer sets we used for each of the virus and what is the target region of the virus where we are going to design the primer set, and what are the expected sizes of the bands for each virus. Sequencing and alignment analysis. Why we are going to do this? After getting the PCR product, the amplicate from the samples, to confirm the amplified fragments. It's belongs to the what we are getting in PCR product or not. This we are doing by sequencing the amplified fragments from PCR products, and alignment analysis using blast service by NCBI, and phylogenetic trees were constructed from multiple alignments using the DNMN software. Results and discussions. For my first objective, we studied here the different isolates obtained from different samples of TVLC viruses infected plants were subjected to PCR analysis using the primer paste designed in study like in table 1, and the results are summarized by DNA of the expected sizes. In the sample 1 is from, this is the sample 1 is from Rehovot. We are going to use 3 sets of primers of different, the TVLCV IL, TVLC mild and TVLCV ES is one first set of primer, second set of primer, third set of primer, and negative and blank. This is the DNA, that marker we are going to use, and this is the expected length size of TVLCV IL we got in this sample. This shows that in this sample the only one virus is infected, like TLCV IL, with the expected band size of 643 base pairs. This is the second sample. We did the same, like we took 3 sets of primers for sample 2, and the first, second and 3 sets of primers in sample and negative 1. Here we got 2 bands of 2 viruses, expected lengths of amplicates like TVLCV IL and TVLCV mild. The TVLCV IL is 643 base pairs and TVLCV mild is 316 base pairs. So you are going to confirm, like you already get the results by the PCR products, but we are going to reconfirm the results that these are the same or not. So what we are going to do here, the amplified fragments we are getting from PCR products are going to sequence, and then alignment analysis was done with sequence virus data available database from gene bank. We are going to get a sequence from gene bank according to Gemini viruses database, according to accession numbers, by online blast technique amplified sequences, and then multiple alignment sequences done by DNMN software. My objective 2 is worldwide geographical distribution of TVLCV associated viruses. How we are doing this is the nucleotide sequences of the viruses are received from the Gemini viruses database according to the accession numbers. I'm going to show in next slide, and sequences are aligned using DNMN software, and then phylogenetic trees were obtained from multiple alignments of nucleotide sequences of virus genomes. This is the table is showing the accession numbers of for different viruses or belongs to the different strains or species. How we are going to sequence the alignments in DNMN software? First we are going to get sequences for each viruses from the database. We are going to align one by one in DNMN software, and then we are going to do multiple align sequence and phylogenetic tree. This is the results for my second objective, that it's showing the phylogenetic relationship of complete genome of different viruses in different parts of the world. I selected India because I am from India and here this is showing the degrees of similarity shared by full length genome of different parts of India where viruses from region are homologies greater than 80%. So these are not belongs to the same strain. These are the different types. If it is 90%, if you can say that this is the specific one, but it is 80%, so it is different strains. It belongs, these all viruses belong to different strains. And this is the phylogenetic tree from the TVLC associated viruses in Eastern Mediterranean basin. Here the degrees of similarity shared by full length genome, the only 2 viruses, that TVLCV, ILF and mild, these are showing 89% of similarities. So this we can say that most similarities are there in between these 2 viruses, though these are very like dominant viruses in these regions, and other than that all are very less percentage, so these are not dominant. These are the traits. according to my research, I didn't get this figure that according to leafcurl at 2010 recent studies, I find this figure that the sequence succession numbers are colored based on TVLCV strains. Here you can see the strain, mild, IL and other. Here the mild is colored and blue and IL is pink, and under different regions of the world like Horn of Africa, Iran, Eastern Mediterranean, Western Mediterranean, North America. Here what we can see, the according to these colors and these colors, which is the major like viruses is spreading all over the world. Here we can see in Asia the majority of virus species is present is TVLCV IL, that Israel, and in Eastern Mediterranean region this strain is saying that is not belongs to EL or mild, is intermediate, because we are not getting now complementary strands of full length of sequences, so it is not belongs to these both. It's called strain. So is in black color. So what we can say, in Eastern Mediterranean region the both are present that EL and mild viruses, and in Asia the major species present is IL, that TVLCV IL, and other regions are like in Iran the other viruses are present. Yet the migration of events interfere using code protein and full genome datasets is also done by leafcurl in 2010. Here how is showing that how the viruses migrated from so many years, like from before 50 years, 40 years and 30 years. Here the lines are showing the age of the migration. So recently from 10 years the viruses are migrating from Asia to the North America, and from 30 to 40 years, here the green color is showing that this region is Middle East. From here to New World is transferring, migrating the viruses, and from Italy is all over the near the places. Like these are showing how the by the survey known TVLCV strains, the TVLCV IL and TVLCV mild have broadest geographical ranges stretching in the whole world from Japan in the east to Spain in the west, and the Indian Ocean, Iceland of Reunion and Australia in the south. This analysis supports the hypothesis that TVLCV IL has been independently introduced to the New World, to the Northern America, once from region around the Eastern Mediterranean and once from Asia. So among the Gemini viruses, the TVLCV EL geographical range is usually vast. As I read in my all the like two objectives, I can conclude that viruses were detected in surveys conducted in Israel are TVLCV IL and mild, were the two dominant viruses found in Israel in infected plants, and TVLCV strains, that TVLCV IL and mild have the broadest geographical ranges stretching in the Old and New World. At last but not least, I like to thank my supervisor Hanuk to give valuable suggestions while doing my research exercise, and also Rina Gorovitz and all my lab mates, especially Iris. And I would like to thank Hebrew University and PEARS Foundation to give such good opportunities to participate in this program. Thank you.
Thank you, Shilpa. Yes, please, questions.
You explained that as different stages of the plant is susceptible for the virus.
Mm-hmm.
At which stage do you think the plant is more susceptible to this virus?
Early stages, like seedling stages.
Seedling stages?
Yeah.
Can you explain the mechanism behind?
Mechanism behind is the virus is going to suck the sap from phloem, and while sucking the phloem, the chlorophyll content will lose, like the carbon, which are the major sources of energy for plant, which are in phloem. So if the fly is going to suck the virus, the sap, then the plant will become dwarf. And also viruses are going to multiplication. So when they were multiplying, They use the all phloem content, but which is energy is present in the plant set, that plants, and the stage is very early stage is plant is going to multiply the cells very rapidly. So that is the stage that which is the virus is multiplication is more.
Salam, please.
You compared virus from different regions, right?
Yes.
Did you see any differences in the virus when they were sequenced?
Yeah. There is the differences. I showed in the phylogenetic tree.
Rina,
This
please.
is virus transmitted by seeds.
Maybe. Sometimes it will happen.
Maybe.
This no? I'm not sure, sorry.
How is this virus transmitted such long distances across the Atlantic?
By whiteflies.
By whiteflies.
Other than
By whiteflies?
Yes.
So the whiteflies can fly such or what? I mean, of course there are winds, but on the way there are so many obstacles. I mean, rain is falling, humidity. I mean, can they make it all the way?
Like when we are transporting the products which we are producing in Israel, like example, to the European countries,
No, the products are very clean.
maybe some cases it will happen,
Okay.
maybe. And major source of transmission is the whiteflies.
All right.
Control
Thanks.
this virus only controlled by the whitefly or another measures to
If you control whiteflies at first, then you can avoid the virus. I mean, entering into the plants, and also the viruses can transfer by soil, like soil-borne diseases.
Then, if it is transmitted by the soil, according to your soil by plant material, how to control that virus?
There is no measure to control viruses still yet.
And which part of the plant is susceptible to the virus?
Phloem, like all the parts, as I said in the first itself, all the parts of the plant is susceptible to viruses.
What is the mode of damage that this virus causes to the plant?
The virus,
How does it interfere with the normal plant development?
it's going to enter to the phloem part of the plant. So in phloem part, it will going to divide the viruses. They are going to utilize the energy from plant part of the phloem part. So when the energies are going to low level in plants, they will dwarf and curl and chlorosis, like losing the color. Those things will happen.
Just due to being in the phloem, or do they damage cells there, or what?
They will damage the cells by sucking the sap from the cells.
The viruses?
Viruses multiplication will use the energy from the plant parts, like phloem parts.
Okay.
Yeah. So the plant will, when the energy source is reduced, then plant will dwarf and stunting and growth will reduce.
Okay, other questions? Irmisrach, and then Gabriel.
Can I ask you what is the overall importance of your study? You are like looking at the distribution of the virus. So what is the use?
Use is when we are going to know that these are the main strains are present in particular region. Like for example, in Israel only TVLCV IL and TVLC mild viruses are present, as we studied here, also by literatures. And also there is a chances of entering other strains from other countries, like TVLCV Sardinia or Cilici, like other viruses. So we can find any other viruses are also entering into the country. We can find by sequencing, or we can produce a particular variety, like resistant varieties for particular virus, which is majorly present in the region.
So you will be looking for resistant varieties depending on what is present.
What is majorly present.
Gabriel, please.
Let's say, well, now that you know about the distribution of the viruses and so on, there's, let's say you go back to India and you get into, say, you get a job into the National Agriculture Organization, something like that. And what actions would you implement in order to avoid the spread of certain virus that is not from India, like in order to not to get into India.
Not to
Or
get
Canada.
into the India.
Exactly. Foreign
Like
virus.
while sequencing, we are going to find means there are some other viruses are entering or not, and we can make a quarantine like not to enter the viruses into the from other sides of the countries.
You mean quarantine to all the products, all the tomatoes that are going to get into India?
Maybe that is not a part. Like the products will not introduce the viruses into the area.
Yes.
The two dominant viruses found in Israel and in another world are equal.
No. The TVLCV, IL and mild, these are closely related, like up to 90%, but not very like perfect, like not same like as the one to each other.
Now tell me something. Sometimes two strains of virus of the same virus, okay, they live side by side in the same field and sometimes in the same plant. Do these different strains of viruses interact with each other? Is there any so-called sexual reproduction in the life cycle of these viruses, or each virus is just by itself multiplying?
Just by themselves they will multiply.
So no interaction, no exchange of genetic
No.
material between the viruses. Other questions? Please. Hello, something? Yes. Yeah.
You talked of there was some identity in your that you said 89% it showed that they were the other percentages were not like similar. So which is the minimum percentage that you can use to show that this is a similar strain?
90%.
90%?
Yes.
Yet it was 89 there? So,
Yeah.
so which means 89 is not
89 also, you can consider that 89, 90.
No, I was asking the minimum.
Minimum is 85.
Okay, please join me in thanking Shilpa. And although we are a little bit ahead of time, and yesterday we took a short break in order to fix this, I will not make it today because yesterday I just lost you. So we shall go on according to the program, although in 5 minutes in advance. And the next speaker will be Filip Slavkovic from Serbia. Filip, please. Oh, okay.
Good morning. My name is Filip Slavkovic, and the title of my research exercise is Effects of Environmental Conditions on Fertilization and Fruit Setting in Date Palm. My work was guided by Dr. Yuval Cohen and Professor Rina Kamenetsky from Volcani Research Center. First of all, I would like to show you the outline of my presentation, which will comprise the short biological and horticultural background of date palms with objectives. Methodology and results will be divided into two approaches, the so-called in vivo and in vitro system with microscopic analysis, and the discussion. In Israel, date palms are commercially grown on the east, all along the Jordan Valley, down to the Arava Valley, and here we see a photograph taken in the south, in the Arava. And if you ever get close to a date palm, you can see that it is a huge plant that consists of an unbranched tree trunk and a crown of leaves at the apex. It is quite large, and it can reach the height of 20 to 23 meters, and also have a radius of about 8 meters. Majul cultivar is grown for its characteristic sweet berries. It's harvested by these huge cranes, whereas while plants are still young, they're often used for ornamental purposes, such as these ones, as we can see here. Botanically, date palms belong to the family Arecaceae or Palmaceae, which is the palm family. All the members of the family are monocotyledonous, meaning that even though they're so huge in size, they have certain properties of very simple and small plants, such as grasses. Namely, they lack secondary thickening of the tree trunk, which makes them quite flexible to strong winds in deserts. Moreover, they cannot be grafted as they lack cambium. Also, these plants are dioecious, meaning that female and male reproductive structures are separated on different individuals. Hence, there are female and male trees. Flower in dates comprises 3 carpels, which are separated, and each carpel has 1 ovule. The major means of pollination in date palms is by wind, so that pollen transmits by wind in a process called anemophily. Upon pollination, or after a pollen grain reaches the female flower and the pistil, it adheres to the very tip of the stigma, as we can see here, the upper style of the upper part of the carpel, and at a favorable moment, it will start germinating. Pollen grain, termed a microspore, has a generative cell that will meiotically divide to form 2 sperm cells. Upon germination, pollen grain elongates into a pollen tube. It penetrates firstly the stigma and the style, as we can see in the scheme 2 and 3, and heads towards the ovule. This is a general fertilization scheme, and I will just note again that in palms we only have 1 ovule. Eventually, pollen tube will reach the so-called megagametophyte, an 8 nuclei structure that has an egg, polar nuclei, synergids, and antipodial cells. Namely, one of the sperm cells will fuse the egg to form the zygote and the embryo, which is the majority of the seed in the future, whereas the other sperm will fuse polar nuclei to give rise to the triploid endosperm. Upon fertilization, only 1 carpel will be fertilized, whereas the other 2 will degenerate. This is what happens under normal conditions. However, when conditions are not optimal or unfavorable, often there is a development of single or triple parthenocarpic fruits. In this case, single or triple or all the carpels will develop to form fruit. In this situation, from horticultural aspect, it is unfavorable as abounding fertilization reduces the quality and marketability of fruits, and we know that more fruits means smaller fruit. So what we tried to do was, by evaluating and approaching biologically and physiologically, we evaluated effects of different temperature regimes on pollination and fertilization. Our main focus were early stages of fruit setting. So what we did is that we measured the amount of germinated pollen grains and elongation rate in 2 systems, in vitro and in vivo. Now I will explain you the methodology. As I said, we calibrated the early fruit setting in date palms by controlling temperature regimes on the trees in the field. Dates were pollinated with normal pollen, and for this particular occasion, special chambers were designed in order to create a specific microclimatic environment around spathes and inflorescences. This is a very new approach and a new project, and it's still it is developing. At this stage, we are only dealing with temperatures. These chambers or temperature units were then installed on the inflorescences, as I said, in the field. Inflorescences were sampled within 6 weeks from pollination 10 times, and bunches were taken to the laboratory to be evaluated and counted. Another important thing is that the time of anthesis, the flowering of dates, is in March and early April. And what I'm showing you with this scheme is the climatic data. So the average minimal temperatures are around 8 to 9 degrees centigrade at this time of the season, and they reach up to around 30 degrees. This is their variation. Here we see the general working scheme of some experiments that were conducted in our lab. I would like you to focus on the one I pointed out and marked yellow. That is the one I contributed. This experiment was based on using 3 different alternating temperatures as treatments. So there was an alternate cold, medium, and warm of 8 to 20, 12 to 25, and 18 to 32 respectively. For control, we used 3 bunches that were pollinated but were not enclosed in bunches, they were only in the field. Moreover, there was another negative control where no pollination was used, but instead bunches were enclosed in bags so that Parthenocarpia will be studied later. 1 bunch was used per replicate, and there were 4 replicates. Now I'm repeating again that this approach is new and it's still being improved. So what I wanted to show you is actually temperature variation inside or within the temperature units or temperature chambers. What we did was that we measured approximately each minute, we measured temperature and averaged it per hour. So we actually here see temperature averages with standard deviations that are marked black in the high temperature unit, medium temperature unit, and low temperature units respectively. The thing I wanted to show you with this is that still there are high variations, especially in low temperature unit, which is not efficient enough when it comes to cooling in summer. When temperatures outside are around 40 degrees, we still need to work on improving cooling capacities and sealing also. Now we come to the first results. We brought bunches to the laboratory and we counted and calculated percentage of normal, parthenocarpic, aborted, non-developed, and drying fruits respectively in response to 3 temperature treatments and the control. We did not find significant difference in percentage of normal or drying fruits in response to these temperatures. However, the percentage of non-developed fruitlets was found in response to the treatments as compared to the control. Moreover, we could report that percentage of parthenocarpic fruitlets was increased under cold temperature treatments, which is in green, as compared to the warm control and the control, the warm treatment and the control. We actually compared means here by using the Tukey-Kramer test for mean comparison. Next results show the variation of fruitlet weight in response to temperatures. The highest fruitlet weight was measured under warm temperature treatment, and it equaled 3.76 grams, which was significantly different as compared to the warm and cold treatment, which were much lower. Also, fruitlet weight under warm temperature treatment was significantly different as compared to the control. We used this one approach in evaluating fertilization, but we wanted to complement it by taking fluorescences to the laboratory and using another approach. So we developed an in vitro assay. We brought spathes or structures containing closed structures with closed inflorescences to the laboratory, And at different times after pollination, we sampled and took inflorescences to be evaluated. Now I will tell you more in details what we did. As I said, inflorescences were brought to the laboratory. They were cut and placed in glass tubes containing liquid medium with sucrose. One of the difficulties we faced at this stage was actually short shelf life of cut flowers. Basically, we're here dealing with cut flowers, which is particularly under higher temperatures had difficulties with fungal contaminations. These inflorescences, as I said, were placed in 4 different rooms with constant temperatures of 15, 20, 25, and 30 degrees respectively. And at different time points after pollination, they were sampled. Namely, inflorescences were cut and placed in these little bottles that contain solution, a fixative solution, which is formalin acetic acid ethanol, or FAA, which fixes the material until it is needed to study. Eventually, these materials, these flowers, at different time points, were taken, they were dissected, stained with aniline blue, and taken to fluorescence microscopy. These preliminary photographs are just to show you what we did. We dissected stigmas, the very tips of carpels. We stained them so that we can detect the greenish-blue fluorescence against the dark background. This way, we can evaluate the level or amount of germination under different temperature treatments. In order to grade, we made a 5-point scale ranging from 0, which was no germination, up to 4, which is high germination. Our results showed that, as we expected, the highest germination was reported under the highest temperature treatment, which was 30 degrees, and they were not inhibited whatsoever under this temperature. These results, as we see, were significantly different, again according to the Tukey-Kremer test, to the treatments of 25 and 20 degrees respectively. Inflorescences kept or incubated under 15 degrees were smaller and stiffer and more difficult to manipulate, but they kept a fair germination rate. Moreover, pollen tube elongation was reported under all 4 temperature treatment that we tested. Under 20 degrees centigrade, pollen tube penetration through the stigma in the upper style was reported 1 day, in samples 1 day after pollination. Also, we detected pollen tube elongation at 15 degrees also, which I did not expect to see. However, it needed some more time. The first time detected was 7 days after pollination. It is slower, but it still occurs. Finally, I can summarize that environmental conditions have significant effect on pollination, fertilization, and early fruit setting in date palms. We developed 2 different systems for evaluation of the fertilization process in date palms, having in mind that each one of them has specific advantages, but also has technical and biological limitations of their own. Namely, we can see that date palms are huge. They cannot be taken to phytotron to be studied. Also, by using our chambers, we are aware that still trunk and leaves are not being covered, and they might be under different environmental and hormonal signaling, which we cannot control by this. That is why we used another approach of in vitro fertilization. So we developed for the first time a protocol for spikelet culturing and fertilization in vitro. which also, as I said, while using cut flowers, we face different limitations, such as more ethylene is released when we cut flowers, so there is more rapid senescence, and there are no leaves. As main conclusions, I would point out that lower temperatures promoted parthenocarpic fruit development in the field. Moreover, pollen tube elongation was reported under all temperatures tested. At 20 degrees, it was seen 1 day after pollination, whereas at 15 degrees it was seen after 7 days. These results that I showed are a start point for the continuation of our research for the next year, where we want to include hormonal effects, which we did not have the opportunity to do at the time being. Thank you very much.
Thank you very much, Filip. Javier, please.
Do you know what is the mechanism of low temperatures? How they affect the parthenocarpy?
What we know is that there is a different development as compared to the normal fruits. Parthenocarpic fruits don't reach ripening. They, at some point before, they stop accumulating sugar, so they're stiffer, they don't reach the color, and they don't reach the degrading stage that normal fruitlets. So perhaps these temperatures are, so perhaps it is much less sensitive to low temperatures as compared to the normal fruitlets.
You said that different temperatures you tested on pollen germination?
Yes.
Tube elongation. So which temperature like affected the germination?
I showed that here we used this grading scale from 0 to 4. So the highest germination rate, or the amount of germinated pollens, was 30 degrees. As we can see, that means were at 30 degrees were 3.41, the highest 1 day at 30 degrees. So this was actually the highest reported. That is what you asked. We did not measure ethylene.
No, but you said that you saw a burst of ethylene and had
I only mentioned it as a limitation of the in vitro approach, because when we have cut flowers, as we know, we did not really, we are aware of it as a limitation, but it is as it is, yeah.
Fatah and Daniel then.
There is also found some problem like incompatibility.
Exactly if there is self-incompatibility.
At this point, no, we did not come across of any.
What?
Sorry, I have to comment. I mean, since it's dioecious, there's no issue of self-incompatibility because you have a male and female. You don't have the same organs on the same plant.
Yeah, we used different parents, so we didn't really try to even induce it. We didn't ever.
What was your control? You said that you had a control that the entire bunch was not pollinated and covered. Is this the control you presented here, or
no? I did
the
not.
Control was just open air, natural conditions.
The one that that we used in the experiment
With
was
the chambers,
was the control that was pollinated. We used pollinated, but not enclosed, so it was just in the field.
Okay.
There was another that was not pollinated, and it was kept for later for parthenocarpic research.
I see.
Yes.
Okay. So, but this control.
It is ongoing. It is ongoing.
This control was was different from all three treatments. At what temperature was it I mean, what was the ambient temperature?
It was varying.
What treatment is it comparable to?
It actually contains, it has the highest variation because the controlled temperature goes from 8 to 9 up to 30, and it is the highest variation of all. It somehow contains all the treatments within. I can show in the slide that I showed, it is actually the ambient or the outdoor temperature. So.
But I mean, what you had 3 temperature regimes
Mm-hmm.
imposed in these chambers. To which of these is more related in temperatures?
It is related to the cold in the mean of its minimum, but it is also related to the warmness. Related to its maximum.
So
it is a very big range.
Okay.
Yes. That is why we also had parthenocarpic difference in control, because it also goes down. But
it's a very good point. Maybe we have to think about other alternations for the next year. Thank you very much. Because maybe we have only let's say 13 degrees between. It was programmed for 13 degrees or 12 degrees between the day-night difference, right? So
Right.
you point out here that in reality these alterations is much bigger.
And this one was very different from all
and
3
we are
treatments.
planning next year design for the next now. So thank you very much, Ruki. This is very good point.
Riba, please. Oh no, I had Daniel on my list. Sorry.
That was my question.
That was your question. I took it. Sorry. Riba.
Okay, in your treatment you put the flower in some chamber. Have you checked whether there is a change in other conditions, maybe in carbon dioxide in that area that can influence the performance?
That might be raised later on. We as I said, this project is in the beginning. We tried with temperatures as a first factor to be evaluated. We're having in mind to use humidity, but these chambers as technically are being adjusted to test different things. So at this time only temperatures, not a CO2 for your question.
I mean, is it not possible to use if you are using controlled environment, why you can't use in the greenhouse or any area as example?
It is possible. This is only what we did. So.
Anjit?
Yes, we know that it is monoecious plant. If you have only female plant.
It is dioecious.
Yeah, sorry, this dioecious plant. Yeah, I was confused. So we have male and female plant different. If we have only female plant, is it possible to fertilize plant by other pollen grains from the other species of the plants?
Of what other plants, for example?
Any other.
Other palm species. Other palm species?
Other plant.
Other plant.
Other plants.
For example.
Hardly. Hardly.
And other palms?
Not hardly. The definition of the botanical species is the 2 botanical species can't be crossed by conventional means. This is classic definition of the botanical species. So probably you can't. Just by maybe genetic manipulations or transformations or whatever, but not by classical means, no.
Now, let me ask specifically about pollination between different species of palms. I mean, can you have some kind of maybe through certain techniques and introduce traits from different species that maybe do not produce any fruit to this one?
So you can. Oh, sorry.
Not you.
I'm sorry. Could you please repeat?
Yes. Can you cross palm, date palm, by different palms in order to introduce traits that are not available in the date palm from other palms that are used maybe for ornamental, I don't know, maybe...
Yes, I believe so.
Other traits?
Yes, we are actually
Say, for example, you found a palm that doesn't produce commercial valid fruit, that has tremendous fruit set under any temperature. Now you want to introduce this trait into the majul, to have a majul fruit that will set the fruit at any temperature. Can you do that?
It probably takes a lot of work to hybridize, to try to do it, but I believe there's...
Biologically, work is something else. We are here to work. You'll have a PhD on this
first
project.
thesis, like PhD.
Okay, first the thesis. Okay, all right. I mean, biologically, is it possible?
For different species.
Yes. Interspecific hybridization in palms. Is it? Have you any idea if it's possible?
I did not read about it, but I can only assume
that.
Yes.
That yes. Yuval, can you comment on this, please?
Yeah, I can. So, palms is a very large diverse family. Most of the palm species will not hybridize with date palm. However, there are other fennec species like the canary palm or the pygmy date palm, which are used for ornamental, and they do crosses. So yes, they can. I don't know what's the efficiency of germination, but some of these trees will germinate and will be a fruit. So to a certain extent.
So even no need for embryo rescue or something like that?
To some extent, no.
The question was if you can propose interspecific hybridization by non-conventional
you can.
means. You can, of
You
course.
can.
Protoplast fusion. You can do many things. But if you can do it by conventional...
No, I was asking about
But you were
pollination
talking about pollination and not
with
tissue
some
culture, right?
Embryo rescue,
Embryo
maybe it
rescue
will
or
work.
something.
Yes,
It is
Yes,
possible
but-
by normal pollination, and some of the seeds will be variable and even bear fruits. So yes.
So is it or was it used throughout the history of palm breeding?
It is not, because
You know, I'm thinking
Yeah.
of field crops and
Because
vegetables breeding. I don't know how.
you need to understand what a breeding project in date palm means. If it takes 10 years from seed to tree, and if it's dioecious, meaning few generations will take 30, 40 years, it's such a long time, so breeding is almost not practical by conventional means.
Mhm.
So it's possible, but not in his PhD terms.
All right. Other questions, please.
I just wanted to confirm what were your findings in the fruit set.
On the fruit set.
Yeah.
So I showed that the major thing was to characterize and see how we can affect to induce or not to, or to inhibit parthenocarpy. In this case, we don't want parthenocarpy. We want normal fruits. So it is important to show that temperatures should be within certain limits to avoid this. So the major finding was that cold or low temperatures would induce parthenocarpy. If I can point it out.
Wait a minute, Ranjit. Any others for questions? So, Ranjit, please.
Is there any effect of parthenocarpy on size or taste of fruits?
Yes, parthenocarpic fruits, as I said, have lower concentration of sugars. Their color is more green, so that means that also they have less pigments, and they're more firm and less tasty.
Size?
They're smaller than normal. Yes. Talking about Majula.
Okay, thank you very much, Filip. All right, and we shall move on to our next speaker, Kumar Lama from Nepal. Kumar, please.
Hello. Good morning, everyone. I'm Kumar Lama from Nepal. Today, I'm going to present on my topic, influence of low temperature on quality and aroma of bell pepper fruit after harvest. My research exercise was supervised by Professor Ali Falik. He is from Volcanic Center. Introducing the bell pepper, it is the most important vegetable crop, widely cultivated throughout the world, and prefers in different colors for consumption, such as yellow, green, and red. It is rich in vitamins, especially vitamin C, and vitamin A, D, and E, and K, and minerals and other phytochemicals and antioxidants. It contains more than 200 volatiles, some of which are responsible for the aroma of bell pepper. For the storage of bell pepper, the optimum temperature for storage is 7 degrees centigrade at 95% relative humidity. Major factors of quality degradation in cold storage are chilling injuries. Here we have two pictures of bell pepper. If we focus in these bell peppers between these two black circles, we see there is pitting. This is due to the chilling injury. This is the typical symptom of chilling injury. Likewise, decay development of fungal mycelia, weight loss, and change in volatiles, which are related to the aroma, are the major quality degradation in cold storage. We have cold quarantine treatment. For the cold quarantine treatment, first we have to hot water range over process at 55 degrees centigrade water temperature for 15 seconds, and then we need to pack in plastic film and we store it for 3 weeks in 1.5 degrees centigrade room temperature. From this cold quarantine treatment and the background of the bell pepper, we set our objective to see the effect of low temperature quality and aroma of bell pepper fruit. I want to focus here low temperature treatment on aroma of bell pepper is we are going to do first time. We had our goal to study the relationship of ultra low temperature treatment and bell pepper fruit aroma. We hope that from our results, this will give the information about the external and internal quality of bell pepper fruit after the cold quarantine treatments, that is 1.5 degrees centigrade for 21 days, which is 3 weeks, and 3 days in 20 degrees centigrade. This simulation represents the cargo journey from the Israel to distant markets, such as Japan or the USA. To conduct our experiment, we had two types of bell peppers, one red and one yellow from the Arava Valley, which we harvested at the 90% coloration stage. Then after we wash in, we hot water rinse brush all these harvested bell peppers, then we pack in plastic film, which is XF100 for the treatment group, and pack in 5 kg market size carton, and we didn't pack for the control group. After the packaging, we store it in low temperature, such as 1.5 degrees centigrade, 4 degrees centigrade, and 7 degrees centigrade for 21 days, and 3 days at 20 degrees centigrade. After the post-storage, we measure the firmness of the fruit by using the tensiometer, and we measure the TSS, total soluble solids of the fruit, by using the refractometers. And we analyze the volatiles by using the GCMS. And additionally, we also did vitamin C test. We observed the green cheek. We calculate the decay percentage, chilling injuries, and finally, we also calculate the chilling injury index of the fruits. I want to go to the results and discussion. First, I want to focus on the external quality of the fruits. We have here in my first table the data from the red bell pepper. I want to focus here in weight loss in all treatments. If we see in general in the XF100 packaging fruits, it has less weight loss trend, but especially focusing on 1.5 degrees centigrade of control and it's treated, it has significantly difference in weight loss. Similarly, in general trend, we find higher decay percentage with increasing the temperature, and we find decreasing of chilling injury and chilling index with decreasing the temperature. If we have low temperature, then there is high chilling injury and chilling index. from this table, I want to focus only on the optimum treatment and our cold quarantine treatment in 1.5 degrees centigrade plus XF100 and 7 degrees centigrade XF100. If we see between these two treatments, there is no significant difference in weight loss, but if we look after the decay percent, it has significantly difference. There is significantly low decay percentage in 1.5 degrees centigrade with XF. Similarly, moving to the yellow bell pepper, if we see in the weight loss, we also find same results as per the as the red bell pepper. There is less weight loss trend in XF100, but if we focus on the 7 degrees centigrade, there is significantly difference with its control at 7 degrees centigrade without XF100. Similar trend of decay percentage also found, that is higher in higher temperature, and also we find similar trend of chilling injury and chilling index with decreasing the temperature in increasing order. Here in yellow bell pepper also, I want to focus on these two treatments, 1.5 degrees centigrade XF and 7 degrees centigrade XF100. And also in decay percentage, we find there is a less decay percentage in 1.5 degrees centigrade XF100. So from these two red and yellow bell pepper results, we can say that we can also store our bell peppers in cold quarantine treatment temperature. Moving to the another most important parameters of external quality of fruits, that is firmness. Here I have to say the more the deformation of the fruits will be more soft. So if we see here in the red bell pepper, which is presenting in red color, the deformation is almost less than 3.5. It means the fruits are quite firm as compared to the yellow, because yellow has higher deformation. It is because when we look in the decay percentage in previous table, in yellow bell pepper has higher decay percentage. So this decay can affect the of deformation, that is firmness of the fruit in yellow bell pepper. Similarly, moving to the green cheek of percentage in the fruits, we find green cheek in 1.5 degrees centigrade and 4 degrees centigrade in both variety, but there is higher green cheek in yellow bell peppers than the red bell pepper. But at 7 degrees centigrade, we didn't find any green cheek. And moving to the biochemical changes results, here in vitamin C and TSS content of the both yellow and red bell pepper fruits, what we see, the trend of vitamin C is we find quite reduced in low temperature, but there is no change in TSS in both yellow and red bell peppers. Focusing on both 1.5 degrees centigrade and 7 degrees centigrade, if we see the vitamin C content in 1.5 degrees centigrade with XF100, it has less as compared to the 7 degrees centigrade at XF100 with both fruit, but there is no change in TSS content of the fruit. Last but not the least, it is the most important part of our research, that is changes in aromatic volatiles of the fruits. Here in the pictures, we see this is the 7 degrees centigrade treatments results of our peak areas of our aromatic volatiles. We find most of the 14 volatile compounds which are responsible for the aroma and have the higher peak areas as compared to the others, more than 200 volatiles, and we select 14 aromatic volatiles. And by comparing these 7 degrees centigrade treatment with 1.5 degrees centigrade treatment, we found that it was reduced. Generally, from this graph, we can say the aroma is reduced. But if we focus on specific compounds which are very important for the aroma of bell pepper, here I want to focus on first in these two compounds, that is 1,6-octadine isomers and pyrazine isomers. I have to say here 1,6-octadine and pyrazine, both are responsible for the green bell pepper aroma, that is green aroma, and pyrazine also responsible for red bell pepper aroma and lettuce aroma. It means the aroma of bell pepper is still we have in 1.5 degrees centigrade. And another most important volatiles which which has major roles in quality of the bell pepper, I want to focus in one pentanol group. In these compounds, here is also we find a reduced in amount, but still present in 1.5 degrees centigrade, and it is mostly responsible for pungency, spicy odor of the bell pepper. So the aroma is reduced, but still we have aroma of the bell pepper when we store in 1.5 degrees centigrade temperature. Moving to the yellow bell peppers, likewise in red bell peppers, we also have had 14 major volatiles with higher peak areas at 7 degrees centigrade. And if we compare these compounds with 1.5 degrees centigrade treatment, what we find, it is generally, if we look after it, it's almost similar. But if we focus here in specific compounds, especially on these three compounds, that is 1,6-octadiene, pyrazine, and 3-cyclohexene group, I already told you octadiene and pyrazine isomers are responsible green bell pepper aroma, and this 3-cyclohexene is also sweet green, responsible for sweet green aroma. And I want to focus here, these green aroma are increasing because we had higher green stick percentage in yellow bell peppers, which we saw in the previous slide. So this is the reasons we had quite higher level of green aroma in our yellow bell pepper fruits. From these results, I want to conclude that hot water rinse over versus treatment in combination with micro-perforated bag, that is XF100 packaging, reduce the chilling injury index, decay, and weight loss. There is no significant reduction in TSS, but vitamin C is reduced with the temperature, with low temperature. And in yellow bell pepper, there is no change in aroma. We don't find any change in aroma, but in the case of red bell pepper, it is less aromatic at 1.5 degrees centigrade storage condition. So finally, I want to say cold quarantine bag treatment, especially 1.5 degrees centigrade, maintains the overall quality of bell pepper fruits. For future direction of research, I want to say test panel, antioxidant test, which is also the most important component of the quality component of the bell pepper fruits, polyphenol test, and also I already in the beginning I focused that we are the first who study the relationship of ultra low temperature treatment and change in volatiles. So we need more data and more experiment for additional seasonal analysis. In the last, I would like to thank Professor Al-Falik, Tamar, PEARS Foundation, Hebrew University, and all my colleagues, and who are also, I would like to thank who are watching me on live. Thank you very much.
Thank you, Kumar. We can take questions now.
I think that the taste was very important here.
Yeah.
And you left it out. You didn't taste your stuff.
Okay.
Whom should we start with? Remisa.
I want to ask you when the storage is affecting vitamin C, right?
Yeah.
So what can we say about that? People are consuming pepper, especially for vitamin C. So what's the implication?
Yeah. What we find here, the vitamin C is reduced with decreasing the temperature. It is due to the reason of, we all know that with the reduction in the temperature, most of the metabolic activity are reduced, so it is due to that reasons. But for the quality of the fruits, we are only consuming only for the vitamin C. Maybe this reduction, I think this not a huge reduction in vitamin C content.
Okay, you found that the heat treatment
Yeah.
influences the chilling injury. do you think if you bring fruit or bed peppers grown under different conditions, maybe the nutrient or any other things, can you expect the same result with this? Or that means, can the growing condition influence the final result?
Sensitivity to chilling injury, you mean?
Yeah, you asked about relating to the growing conditions and chilling injury, right? We have, especially focusing on the chilling injury, for inhibition of chilling injury, there is presence of heat-sucking proteins for inhibition of chilling injury, and it is induced by the hot water rinse over process treatment. One reason is that, and we cannot say we cannot say that the bell pepper which we perform the experiment and which we get same results in the bell pepper of Israel, and we will get the same results in other countries like which are growing in different environmental conditions. So it is we are only I want to say that only it is reliable and it is good only for the bell peppers which are grown in Mediterranean regions, especially in Israel.
We can grow under different,
No, but you check
conditions.
it in different months, right?
Yeah.
So pre-harvest was in different months, right?
Right.
Right. So pre-harvest conditions were different, right?
Yeah.
Was the difference between months and
Right.
So a general question: if pre-harvest conditions will affect post-harvest conditions?
Growth conditions.
Pre-harvest
Yeah,
is
we
growth.
can say growth
Yes.
condition or pre-harvest condition. Let's say you applied for example, during
Yes.
the growing for one grown here, 10 milligram nitrogen, for the other you may provide 100 milligram. Can
Yes.
this have effect on the final result?
I wouldn't go to nitrogen because he didn't test it. But
No.
let me ask if you would harvest fruit in February versus in May, okay? And you don't treat it with hot water, etc. But you
Okay.
just put it into the storage. Do you expect them to have a different sensitivity to chilling injury?
Yeah, I have answer for these questions. With decreasing, if we harvest in April, and if we harvest in January or February, in April in the last harvest, our quality of the fruits is external quality of fruits is decreasing. So it's obviously suffering from more chilling injury, and it also affects on most of the external quality of the fruits. Yeah, that's like all those fruits. If you harvest in the last, the fruit external quality is not good. In the previous harvest.
No. It's correct, but let us not stick to this point of the time of the season. Okay, let me give another example. You harvest on the same day, from one greenhouse in the Arava Valley and one greenhouse in the Golan Heights, which is much cooler temperatures over there.
Okay.
Okay, will you expect differences in response to chilling, chilling resistance, chilling injury susceptibility or resistance?
Between these two places
Two
of
different
harvest.
conditions, the same cultivar, the same treatments, the same fertilizer, the same everything, just different locations, different temperatures, or even in the Arava, two greenhouses, one is cooled, one is not cooled.
I don't think it will affect within these regions and within the same species.
Okay.
In your conclusion, you mentioned the total soluble soil is not there is no significant difference.
Please speak loudly and shortly. Question, just question, please.
Okay. On the conclusion, you mentioned TSS was not significant, but you observed a difference in the vitamin C, right? How come the TSS was not significant, but the vitamin C is low, it was going down under ultra low oxygen? They are related, right?
Relationship between what you ask.
Disease in vitamin C.
I don't think that the vitamin, if vitamin C is reduction, and there is the release also needs to reduce this total sugars soluble. So it's these two are basically different component.
Vitamin C is organic acid.
Yeah, ascorbic acid. But it's I think it's due to the region of cold temperature. In vitamin C, it affects more, and then the TSS.
Salam, please. Yeah. What role do you think temperature is playing on the changes in aroma? Like if you have low temperature, you have no. Yeah. What role?
Okay. Mostly there is changes in red bell peppers. What I found in literature review in red bell pepper, especially in red bell pepper, not in yellow bell pepper, the lipoxygenase that is we call LUX. That LUX cycle, when it ruptures the cell, it reacts with fatty acids and oxygens, which produce hypoperoxides, and then these hypoperoxides creates it reacts with enzymes and it creates linoleic acids, which are most responsible for the precursor of scents in bell pepper, red bell peppers, and especially in hexanol groups and its isomer. And hexanol in bell peppers, they are responsible for grassy smell, that is greenery smell. So in low temperature, it will inhibit. So there is reduction of volatile compounds in bell pepper, red bell pepper.
In the general metabolic chain, when the synthesis of these volatiles occur, before harvest, during growth stage, or approximately at harvest, or during storage period?
Thank you, madam, for good question. The changes in formation and changes of these volatiles occurs with maturity of the fruits. You cannot get all these volatiles when it is in immature stage, I mean green stage. Red bell pepper, or in the beginning it's green. It has different, it has more hexanol groups of volatiles. By maturity, when it turns into the red color or yellow bell pepper in green color, these hexanol are losing. I mean, these are reducing with maturity, and with increasing the temperature, I mean maturity or ripening, all those volatile compounds which are responsible for the pungency and spicy, they are increasing. Generally, if we break the two bell peppers, which are immature and which are, we in a purple color, we find two different smells. Obviously, we get in green more grassy, greeny smell, and in red bell pepper with different smells.
During your study, you measure some specific stage of development, post-harvest development?
No, we just harvest fruits at nearly 90% coloration stage.
Can you breed pepper that will be resistant to transportation? You don't have to chill it. That will be the solution, actually. Can you do that? Do you see any way how to do it?
Sorry, sir. Can you?
I say that the problem is transportation. What happens to the fruit during transportation? Can you breed and select pepper that will be stable, resistant to transportation? You don't have to chill it. Just put it in the boat, and that's it.
Sorry, I don't have that much idea about the breeding, but for the transportation, we have these options: cold quarantine treatment. Especially cold quarantine treatment is developed for the prevention of Mediterranean fruit fly, because Japan and US, these countries are free from Mediterranean fruit fly, and we developed this in the 1.5 degrees centigrade. We kill eggs, larva 1, larva 2, and larva 3 stages of all Mediterranean fruit fly, but we don't know about what's the quality of bell pepper. Maybe the bell pepper will be tasteless, aromaless. So we doing this research here.
So for example, tomato shelf life was very low, 40 years ago. It was 1 day, 2 days, and then it rots. Then they found a gene, okay, 2 genes actually, that stabilize the tomato. Now you can keep it in your cupboard for 1 month, nothing will happen. Can you imagine something in pepper? Something like this in pepper?
Maybe if we get that type of bell pepper, then no need to do these things. That's good.
From where did you take the genes? Any idea? Just you think about it, or you never thought about it?
No, I didn't focus about the other part for the genes which are related to the strengthening or longevity of the post-harvest life. I just mostly focus on the aroma part, which are typical aroma for bell pepper.
Okay, thank you, Kumar. So, just I would say a kind of intermediate summary, and immediately you will understand why I say intermediate summary. We've heard until now, yesterday and today, 15 presentations of our students presenting the research exercises they conducted under the supervision of different researchers, either here at the Faculty of Agriculture or at the Vulcani Center, the ARO. You can get the impression of how diverse are these topics, and each of them is interesting by itself, and the students did most of them, or even all of them, very good job in conducting the research and presenting the results. We still have ahead of us 2 more presentations, which unfortunately I cannot invite our viewers over the web to watch, because these are 2 studies that are confidential and they will not be broadcasted. But we will continue here after lunch with the 2 last presentations of our students. And I apologize to these watchers over the web that they will not be able to watch it. So I would like to thank all the speakers until this point, and of course the PEARS Foundation for supporting this webinar. Thank you very much.